Hi everybody. Today we're going to learn how to make a pH indicator for the CO2 lab for the respiratory section in your lab packet. One of the ways to measure carbon dioxide amount is to measure acid as it forms in water, because when you blow bubbles in water, you're blowing carbon dioxide into that container. And when carbon dioxide and water mix, they form carbonic acid. Carbonic acid is a weak acid, so it comes apart into the hydrogen ion and bicarbonate. You should remember this from the bicarbonate pathway. But the presence of hydrogen ion makes the solution acidic. So today you guys are going to blow bubbles into water after rest, after exercise, and after hyperventilation. And the presence of acid is something that we can measure. Now at home, because you don't have a pH meter and you may not have litmus paper, what we're going to do is make something that measures pH. And what you use is red cabbage. Okay, and you can buy these for $1 at Meijer. And all you do is put your red cabbage in a blender and add water. And I have 2 cups of water in here, but pretty much it doesn't really matter. So you just blend up your cabbage. Shake it up a little bit, but you're going to get basically purple juice from your blended cabbage. And so you take it out and you put a filter. You can use cheesecloth or even a piece of gauze. I'm going to try it today even just without that, because the filter itself is going to catch most of the big chunkies, and it doesn't even really matter. Okay, so now you have purple cabbage juice in this container, and notice the color of it is kind of a dark purple, and the pH of that solution, it's just my tap water, should be pretty close to neutral. So a neutral pH shows up basically purple. So I'm going to put a little bit of this into these glasses, and I'm going to add some solutions and show you what happens when you change pH. So for example, vinegar is an acid, and if you put just a touch of vinegar into this glass, notice the solution changes to pink, because the pH indicator in the red cabbage juice goes violet to pink to red as pH drops. So vinegar is acidic. It got down towards the pink end. Bleach is basic, and a basic solution is pH above 7. And so if you add just a little bit of bleach here, notice it turns green, blue, and then, or it goes from blue to green to yellow as it gets more basic. Bleach has a pH of like 9 or 10, so it's getting, all the way to yellow practically. And vinegar has a pH of 3 or 4, I think, so you get down towards the acidic end. So now you have made a pH indicator, and it's kind of fun. You can add, a little bit of the purple grape juice to any solution and get a rough idea of pH. So in your lab, what you're going to do is you're going to have 3 containers, and generally it's better if your containers are clear, because you want to be able to tell the color. And what you should do is put the exact same amount of the pH indicator into each glass. So I'm going to use 1/4 cup because my glasses are 1/2 cup in size. And you could use more. You can dilute this pH indicator. If you didn't get quite enough, just add more water. You just want to make sure you have the same amount, the same concentration in each glass or in each of your containers. Spilled a little bit of that. Okay, so what you're going to do So you're going to have your 3 glasses, and you have to have somebody willing to play along here, and it's kind of nice to have a second person because you're going to time this. So what you're going to do is a person at rest, or the first time that they blow bubbles, they haven't been running around or anything, and you're going to have that person, you, whomever, blow bubbles into that container for 1 minute. Now, if they have to stop and take a breath, just pause your timer and then restart it. But you want to go exactly 1 minute. So if you have a timer, you can do this on your phones, of course. Set up the timer so that it's set for 1 minute, and you can hit pause if the person needs to stop and take a breath. Okay, so I'm going to set my timer for 1 minute, and I'm going to hit start, and I'm going to blow bubbles into the solution. Okay, so I have blown carbon dioxide into that water for 1 minute. Remember the equation. Carbon dioxide and water is going to form acid. And remember, our pH indicator goes from bluish purple to fuchsia to pink as it gets more acid. And you can see, if you compare the one I haven't blown into yet and the one that I have blown into for a minute, is now more purple. Okay? It has become more acidic because some carbonic acid was formed spontaneously. In our body, carbonic acid is formed much more rapidly because there's an enzyme, carbonic anhydrase, in our red blood cells that make this reaction go faster, so the bicarbonate pathway occurs much quicker in the red blood cells than it does when we just blow bubbles in there. There's no enzyme. Okay, so some of the carbon dioxide and the water formed carbonic acid, and you got a slightly more acidic condition. Your job in this lab then is to hyperventilate for 1 minute, don't pass out, and then blow bubbles for a minute into glass number 2. And you're going to look at the color. And then you need to exercise, do something, jumping jacks, run around, go up and down steps, whatever you need to do, and then blow bubbles into the third one for exactly 1 minute. You're going to record your results. Is it going to be more acidic than this solution, or is it going to be more basic than the at-rest solution? You should be able to predict what happens ahead of time, do it, and then you're going to put your results, describe the color of your solutions in your lab packet, and then you're going to post your results in Teams, and we'll talk about it as a class. So that's how to make a pH indicator and do your CO2 lab.